Cathy Ann Miller joins us tonight from Santa Rosa, where she works in Berkeley. She was just saying she commutes back and forth, and is a curator of the algae in the herbarium at UC Berkeley, which I would love to go and visit sometime. That would be a fun field trip. So without any further babble from me, I introduce to you Cathy Ann Miller, and thank you again for being here.
Thank you. Well, I'm very happy to be here because I always like a chance to talk about seaweed, especially to people interested in seaweed. Those beautiful, fascinating, inscrutable, and wonderful organisms that are often overlooked in the ocean when you're looking for things that are more mobile. So I want to give you a little glimpse into my world of seaweeds, which is my own. This is where I work. There are 2 herbaria, plant museums in this building at Berkeley, at Cal. One is the University Herbarium, and that's the one I work on. That has plants from all over the world, hundreds of thousands of specimens, including algae, fungi, lichens, and flowering plants. The other herbarium in this building is the Jepson Herbarium, which has California flowering plants. So I am representing the University Herbarium with you tonight. The herbarium was founded in 1895 by William Albert Setchell, one of my heroes. He was a phycologist trained on the East Coast at Harvard and Yale, came to Berkeley when he was a young man, earliest 30s, and started the herbarium. So we seaweed people were there on the ground floor. He was joined later by Nathaniel Lyon Gardner, another incredible worker. And it's really thanks to these 2 people that we know as much as we do about seaweeds. They were the foundation of our knowledge of California seaweeds. This is the herbarium, kind of a forbidding looking place, kind of industrial looking, but inside these cases is a world of natural history. These are the seaweeds in these aqua-colored cases. All natural history museums today have grown from the cabinets of curiosity of the 17th and 18th centuries into the 19th centuries. These were places where people collected nature, pieces of nature, and their own fantasies about nature. It's the interface between the human curiosity and the natural world. We'll never give that up. It's as much about us as it is about plants and animals. Here's a very famous lithograph of Algarum vegetatio, the vegetation of algae. This is Postels and Ruprecht, 2 Russian explorers. One was an artist, one was a phycologist, a seaweedologist. So this is 1840. They were on a grand expedition across Alaska, from Russia, across the Aleutians to Alaska. And here you can see the Russian soldiers collecting seaweeds in great bundles. Here is the Alaskan shoreline. And then below the surface, you see the world of the seaweeds. Maybe you can recognize Nereocystis, the bull kelp right here, and the other kelps and understory red algae. This was in 1840. This was long before Setchell and Gardner started studying California. All of what we knew about seaweeds on our coast were studied by people either on the East Coast of the US, in other countries, and especially in Europe, where the great universities were. So it wasn't until Cal got its own herbarium that California really joined the effort to study seaweeds. So my life in seaweeds has been a long one. 45 years I've been at this. The way I approached learning about seaweeds, I took my first course at Bodega Marine Lab in 1976, and I was hooked. And what I did for the next decades was collect and look at seaweeds. Luckily, I was connected with the herbarium very early, so I had something to do with my collections. But I collected in the intertitle, I collected in the subtitle. No seaweed was too small to study. And what I've been doing for so many years in the study of the systematics of seaweeds, their relationship to each other, is take my plant press to the shore and make specimens. So this is what a specimen looks like, pressed with a label, with the name, where it was found, when it was found, and who found it. So specimens have a magic of their own. You look underwater and you see this creature. You pull it out onto the boat, and this is what it looks like. You put it in a plant press, and it becomes two-dimensional, but still retains the tissue of the original and the form of the original. In fact, that tissue is so intact that it can be sectioned and the anatomy can be looked at with a microscope. Plants have cell walls. They come in very handy for making good specimens. Don't try this with an animal. It's squishy. Sometimes we knew things only because they washed ashore. The great elk kelp, Pelagophycus. This is William Albert Setchell in his youth. This thing washed ashore, was collected by Mr. Johnston here, and they made this specimen. Doesn't really do it justice, does it? That's what it looks like in nature. One of my favorite species. This beautiful thing is not a California seaweed, but I'd always admired this lithograph by Harvey, who was one of the famous Irish phycologists in the early 1800s, this beautiful detailed drawing. Then a friend of mine sent me this micrograph through the microscope, and you can see the net-like structure of the blades. You can even see these structures that hold spores here. And it wasn't until, I think it was 2014, that I actually saw this thing alive in the Philippines. And because I had seen these photographs, I recognized it immediately and felt like a puzzle piece had been added to my life. So what we do with these specimens in the herbarium is store them forever. And since about 2012, we've been imaging them and databasing them so that you don't have to come and visit the herbarium to see these specimens. So this is an example of Pelagophycus, the elk kelp. These are specimens of that great beast. And the website here is our public portal. Anyone can come and look at specimens. It's open to the public. This is very useful for working at home too. But I need to tell you that the seaweeds are wily, they are tricky, and we will be studying them forever because of the variation in their forms. For example, this is a Japanese seaweed. There were once 5 species in Acrosorium described. And here are some of them illustrated. They are all one species, folks. Their form is variable depending on the environment, and that's what makes seaweeds hard. You can put a human in various environments, and they do look different, but you always know you're a human, and you can pretty much get to us to species. But you can see the variation in form of this single species. Conversely, these 6 pressed specimens look very similar. They're broad, flat, red blades. They are all different species. Very difficult sometimes to distinguish the flat red blades. So as I say, the weeds are wily. It's worth a lifetime studying them. So let me just summarize why we make specimens, why we have museums, the power of these specimens. They are a legacy for the future. The world is changing quickly, and we need to show what we have now, what we had in the past, to those who come in the future. Not just a list of names, not just an idea, but the actual real things pressed on paper, real specimens. Specimens provide hard evidence of what lived in our ocean. Without a voucher of our observations, a name is just a rumor. Names change, the plants do not. So we need to preserve the plants themselves for that future. To have these specimens is a huge assistance with species identification. That's one of the ways I learned, was collecting, using a book that I'll introduce you to, but also comparing with specimens in the herbarium. Luckily, I was an undergraduate and a graduate student in the herbarium. I worked as a research assistant there, so I had access to the collections and learned how to use them. The specimens in the herbarium are also records of geographic extent. So the more widely you collect, the more you can see the range of a species, its northern range, its southern range, and watch how those ranges may expand or shrink with time. Even in my own time, I've seen changes in species ranges, my own lifetime. Nomenclature is the science and the art of naming things and passing those names along in a logical and rational way. Names are anchored to specimens. When you describe a specimen, you indicate a type specimen, the one that anchors that name to a specimen, and everything else is compared to that specimen. That sounds kind of tricky because we know that species have lots of different forms. How could one thing represent a species? Well, these specimens are treasure houses of DNA. And we now use DNA sequencing to identify species, compare them to type specimens. We have 2,500, something like that, type specimens in the herbarium. And my colleagues are magicians at getting the DNA out of these very old, sometimes hundreds of years old specimens, and allowing us to compare it with specimens in the ocean and understand species ranges and relationships. So let's talk about my favorite thing, the California seaweed flora. That is my research specialty. We're going to look at California. So we are roughly here. I'm roughly here. And we're going to look at how seaweeds are distributed throughout the state. Notice Point Conception here, the bump just above Santa Barbara, because that is a huge break in seawater temperature. Here's the West Coast from Alaska through British Columbia, Washington, Oregon, and California. These colors are cold water. These colors are warm water. So you can see the gradient as you move down the coast. Cold, 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 NorCal, cold. San Francisco Bay, we get a transition into what we call the Central Coast. There's Point Conception. And look at that break. Suddenly the water is much warmer, and down Baja California, Mexico, into the tropics. You see this gradient. Look a little bit closer at California here at sea surface temperature. And again, this is in September. This was 2016 when NOAA made this chart. Cold, 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 warmer, 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 Point Conception, and something very, very different south of Point Conception. So what causes this gradient in sea surface temperature. It's all about currents. The California current coming down with cold water from the north, warm water from Hawaii, Central Pacific coming in from the west. And then these red arrows are warm water coming up from the tropics, up the coast, and mixing here in this Southern California bight, just south of Point Conception. So you can see on this map, the cold water current coming down, and notice it goes a little bit beyond Point Conception. These are the California Channel Islands. Notice all of them, each one is in a different temperature regime. San Miguel Island is sometimes as cold as Monterey. Anacapa Island can be as warm as Baja, or as Catalina and San Clemente Island, the southernmost islands that are bathed by this warm current coming up. But notice too, there are some cool areas on this map. Those are upwelling areas where cold, deep ocean water is upwelled up along the coast as the wind blows the surface waters to the west. And it's that upwelling that allows the kelps to extend their life down into these warmer areas. You can see upwellings here along the coast here and here in otherwise warm water. Upwelling is seasonal, but it brings nutrients to places that are more generally warm. So Northern California and further north is a cold temperate biogeographic province, an area with communities of plants and animals that evolved together. The kelps evolved in the North Pacific, and the cold temperate biogeographic province is rich in kelp biodiversity. You probably recognize this one, Postelsia palmiformis, the northern palm. This is what it looks like underwater in that cold water. Lots of red seaweeds with intense, deep pigmentation. All of these in the background too. There are crustose corallines and articulated corallines, those seaweeds with calcium carbonate in their cell walls, very important structural elements. And here are just a few of these northern species: Gloriopelus, Friella, Caliarthron. All of these are typical of cold water habitats. So now let's go south of Point Conception, where the water is warmer, in Southern California. Pretty different looking, isn't it? This was during a warm water time. It wasn't the warm blob, but it was during a warm summer in Southern California. You can see that the kelp is eroding a little bit. It's not as healthy as it used to be because the water is warm, not enough nutrients to support that huge body. Instead, you see a lot of low coverage of browns, corallines, reds, but notice the reds aren't as conspicuous. Their pigments aren't as deep as in the cold water. So here are some typical seaweeds from Southern California. I did a lot of work in the Southern California Channel Islands, which are pretty much untouched. No, nothing's untouched anymore. That's wrong. Far less touched than the Southern California coastline. So again, you see small plants, light colors. There's articulated corallines, jointed corallines. There are plenty of crustose corallines growing beneath them, and a lot of diversity, but very low stature. So the book that I learned from was Marine Algae of California, published in 1976. By coincidence, the year I started to study seaweeds. This is my book over here. Oops, I didn't mean to do that. Can I go back? Yeah, no. Excuse me, everyone. I have to figure out how to go backwards here. But I'm not doing it. Hold on a sec.
Your arrow is all the way to the left of your screen on the lower part.
Oh, I see. Thank you, Sarah. Right on. Marine Algae of California was written by Isabella Abbott and George Hollenberg, and I was lucky enough to meet Isabella Abbott, though Hollenberg died quite a long time ago. This book reported 669 species. Now we know that 500 of those names have changed since the publication of that book. See what I mean about names changing? They change because we learn more about relationships. We've also added 130 taxa because of discovery, looking around, naming new things. And some species have been introduced to California, 35, from other oceans. And I'll talk about this a little bit more. So I said that before Setchell and Gardner started Cal's herbarium and started studying the seaweeds, there was very little known. These 361 California seaweed species with type localities, that is, that's where the type specimen was collected in California. And many of these are Setchell and Gardner's. And of course, it's been 128 years since they started the herbarium. Many other people have described new species. 60 California seaweed species were described from Mexico. And that makes sense because the coastline continues down and borders are arbitrary for seaweeds. 35 California seaweed species have type localities in Asia, primarily in Japan. And in fact, we, as you'll see, share species with Japan. But some species that were once thought of in common, California and Japan, in fact, are not. And I'll describe that. And then whopping 250 California seaweed
It's
species.
19 hours.
Oh, quiet. Were described from other parts of the world. And we're finding that some of these species do not really occur in California. They just look like species that occur in other parts of the world. So we're trying to figure out how many really do we share with the world? How many are cosmopolitan? So here's some examples of species that actually do occur on both sides of the Pacific or worldwide. And almost all of these are warm temperate because when the world was young, the belt around the equator was fairly continuous when the continents had not moved to where they are today. And the tropical seaweeds were blown with currents around the world. Many tropical seaweeds are cosmopolitan. So these are all California species that are found in Japan, for example, or Korea. However, sometimes species we thought we had in common with Japan are not. Pykea California, described from Golden Gate, was thought to occur in Japan, but it turned out that a lookalike that they then named Pykea yoshizaki is the Japanese species. So in fact, Pykea California is not on both sides of the Pacific. Instead, this cryptic or hidden species turns out to be the same, very different, even though it was misidentified as Pykea California. But interestingly, Pykea California has been reported in Great Britain. How it got there? Some people think that the early flying airplanes with big pontoons brought it. We're not sure. Many species evolved here and are unique to this coast. They aren't shared in other parts of the world. Once more, Neochondria nidifica was called Chondria nidifica, and it was also reported from Japan, but the Japanese one is maybe Chondria nidifica, but ours is Neochondria nidifica. It was described from near San Diego. So again, many species evolved here and have not in any way spread to other parts of the world. Rhodamina Californica is not in Peru. In fact, this shows you lots of dichotomously branched flat red blades. They've had many names over the years, but it turns out these are all one name, and that's Rhodamina corallina from Peru. This one with the star was identified and was as part of their flora, Rhodamina californica. It is not. It is only in California. Actually, it's been reported all over the world, but hasn't been tested. So species ranges have expanded over evolutionary time, long periods of time. I'm going to give you an example of Gloriopelus Furcata. It is distributed from Kamchatka, Russia, Eastern Russia, through Southern California, huge range down the entire coast. And I thought, in fact, that the Southern California specimens, which are much smaller than the northern ones, might be a different species. That's a long road to hoe. But especially since most of the species in this genus occur in Asia. But it turns out it's one species all the way down the coast, and that this species over evolutionary time has crept down the coast by dispersal all the way to Southern California. In my own lifetime, I've seen species expanding from the south into California waters. Warming water trends promote this, of course. This is Asparagopsis Taxiformis. This is the seaweed that they're experimenting with feeding to cows to lower methane eruptions from cow digestion. So it's been very interesting. There's a lot of commercial interest in this species. Very common throughout the tropics. It was described from Alexandria, Egypt, but very, very rare in California when I first started diving down there. Now it's becoming more and more common. It comes in two forms, a haploid form and a diploid form that look different, but they're the same species, different parts of the same life history. And now we're seeing more and more of it as time goes on in California. It will never get above Point Conception. It is a warm water seaweed, but still its range is expanding from Mexico. Here are some non-native seaweeds that have made themselves at home in California. Sargassum muticum, the wireweed, sometimes it's called, introduced British Columbia in 1950s, made it down to Southern California by 1970. So in about 20 years, it marched from an single introduction from oyster spats at an oyster farm all the way down to Southern California, where it has joined the communities, the seaweed communities. Lomenteria hacadidensis is from Japan. It's in Marine Algae of California in 1976. It was listed as native, but now we know it is not native. That has been introduced sometime, sneaked in here, we weren't watching. Similarly, Gracilaria vermiculophila, a long wormy thing, grows in sandy habitats. We don't really know how long it's been here. It's also from Asia, but we have gotten DNA out of specimens from the late 1800s and have found it was there then. So it probably came very, very early, either by transport on ships, by tsunami debris, who knows how it got here. But it is, in fact, in almost every cove in California. Species that have been introduced in my lifetime that I was there to observe the introduction include Ondaria panatophida, which is one of the most edible seaweeds in Asia, the miso soup favorite, showed up on, let's see, where was it first? The mainland coast of California, but I found it on Catalina Island at about the same time they found it on the mainland coast, probably brought by shipping, possibly as microforms on a ship's hull. It has expanded to Santa Barbara Harbor, Monterey Bay, San Francisco, and it hasn't gotten any further north, to my knowledge, than San Francisco Bay, but it may continue to spread. So the Sargassum horneri, now forming huge meadows in Southern California, first observed in 2007, Long Beach Harbor. Someone brought me a specimen and said, "What's this?" And I said, "I don't know. That's not Californian." But we had a visiting phycologist from Japan who said, "I know what that is. That's Sargassum horneri." So both of these were discovered in places where we knew they did not exist in this millennium. And they are continuing to come. Budimo cylindricus is this funny little thing, also in Japan and Korea. I knew it was an unusual plant. I collaborated with some Japanese phycologists, and we demonstrated that it is a Japanese species that is new to California. When it arrived, we don't know. And now there's a paper right in press. We just discovered that Tynocladia crassa is a Japanese species that fairly recently got to California, but we don't know when. These both, as you see, grow in sandy habitats. That's not the kind of place where people look for seaweeds so much. And it's a good place for non-native seaweeds to settle without competition from the natives. So these are growing on pebbles and shells. So where were California seaweeds described? I sort of have a different feeling about this after my experience with all these non-natives and these broadly dispersed species. This native status of many of our California species need to be investigated. We don't even know really what our natives are. We're sure of the kelps because we know they evolved here, but the other ones we're learning. So in summary, you can see that our West Coast seaweed flora is a mosaic of native species that evolved here, of naturalized species that have come from somewhere else, been introduced, but have joined the communities, and these recently introduced species that aren't completely worked into our communities. They're still either only in harbors or in habitats that don't have a lot of native species. Sargassum horneri is an exception. It is actually competing with Macrocystis, the giant kelp in Southern California. The ranges of species change over various time scales, from evolutionary time scales to decades. Asparagopsis has changed its range in the last few decades. And I predict that other seaweeds are going to be changing their ranges as a result of our warming and acidifying oceans. Southern California species are going to be able to round Point Conception to the north as the California current weakens with climate change, allowing that warm countercurrent to move further up the coast. We know this is possible because during El Nino years, Southern California fish swim up to Monterey Bay, San Francisco Bay, and then rarely persist long as colder waters come back. But as warmer waters persist in our northern coast, we're going to see species ranges changing and see more southern species. So climate change is a huge driver of species range changes, just as it was probably a driver of the evolution of seaweeds in ocean basins. But humans are enablers because we move species around, just as we've done in the terrestrial zone, where we brought delicious species with us as we move to other continents. We are moving things in our ballast water of ships, on the hulls of ships, on the hulls of sailboats and pleasure boats going from port to port along the coast. We are moving species, and they do the best they can. They survive or not. And finally, there's been a revolution in the study of these things because molecular studies can determine where species originate and how they've dispersed over time. Because the DNA evidence is deeper than the morphological or form evidence and helps us determine things that we could not see before. So obviously I'm going to keep doing this because so many puzzles remain, and each of these species has its own story. It's very difficult to generalize because this one did it, these other ones will do it too. Each species has its own trajectory in time and space and sings its own song. And that's another reason that keeps me keeping on. So when you go down to the seashore, I hope what I've told you tonight gives you some insight into the complexity of these seaweed communities. I'm speaking as a systematist, a taxonomist who's interested in who is who, but of course, as an ecologist, you wonder, how do these communities form, stabilize? How resilient are they? And how is that dependent on the individual species? So the foundation of our knowledge of species, I've always said, is who. And then all the questions and all the answers arise from that. Thank you. I'd love to hear some questions.
Cool. Well, I think that you've just gotten us all very excited.
I hope so. I mean, you're talking to a fanatic here.
I love it. I love it. Let's see. I think I can look at the chat a little bit. There was definitely some interest in the website where we can go and explore and look around. So Trey will post that up for
Good.
Folks who want to see and explore.
I know it went by very quickly, but I encourage everyone to take a look. I know I name names, and many people don't know the scientific names, but I hope that will change a little. I'd like to teach people more of the scientific names because that is the entryway into this search. But you can also put a collector or a place. So you could put any place on the coast in the box for locality and then see what seaweeds come up. My only hint for that is to hit grid. You'll see what I mean when you're in the program. Grid will give you images, not just words. And what you really want is images.
Right.
It's an incredible database. Beautiful. I think we have we're getting close to 90,000 California seaweeds or West Coast seaweeds in it.
Wow.
Big and wonderful.
Wow. Fantastic. Let's see. I know I had one question about the Macrocystis in this area because we do a beach survey up here, and we have many of the survey folks are here on the call and have asked me questions about what different seaweeds are, and I kind of stop short of 5 or something like that. But the Macrocystis that we're seeing here, there seemed to be some interest into its presence on the beaches, and we see it at Van Dam, and then we see it maybe dispersed a little bit in south and north, but that's beach cast, not necessarily growing. I was just curious about its presence up here along the Mendocino coast.
Well, the northern Macrocystis used to be called a separate species. it was intertidal mostly, or very, very shallow subtidal. It was called Macrocystis integrifolia. But now we know it's only one species of Macrocystis on our coast, and it goes all the way up to Alaska. The herring roe on kelp industry in Alaska is Macrocystis kelp. So the funny thing is its distribution it's not continuous.
Right.
It's spotty. So we don't really know the relationship of those northern populations to the southern ones. It's probably a case of dispersal. It probably happened a long time ago. So it originated in the north. It found a really good home in the south. There's much more Macrocystis in the south, but we still see pockets of it all along the north coast.
Okay, so pockets. So we're documenting that and writing it on our survey forms, so,
like
which is great.
Book documentation.
Yeah.
Interested in your beach walk.
Yeah, people love to dive in and talk about what they're finding. Let me see if there's anything in the chat.
I have a question. Can I ask?
Jennifer's been waiting.
Hi. Yeah. Thank you so much, Cathy. And that was super interesting. I also have a Macrocystis question. So I live in Monterey, and I recently got back from diving in South Georgia and Patagonia and Chile. And I'm curious, is that the same species of Macrocystis? Like, I've read a bunch of papers saying, like, do we have the genetics to know if it actually is like a monotypic genus, or?
There might be a different one in Chile. I'd have to review that a little bit, but at some point, Macrocystis jumped the equator.
Do you know how that happened?
The ocean was different back then.
Okay.
Yeah, that's what's hard for us to. We humans are kind of embedded in our own time, but times were different, and the equator was always been the band around the Earth, but currents were different, continents were different, and the kelps are ancient, millions of years old. So at some point, they oozed across the equator, and I haven't reviewed this recently. I wish I could answer you more completely, but it's the same genus for sure. Other species have been described down there. I know Macrocystis pyrifera, our species, occurs in the Southern Hemisphere. How wonderful to dive at South Georgia.
Oh my gosh, it was the most amazing kelp forest I've ever seen in my life. But I was wondering, like, would you know by looking at it if it's the same? It would probably have to be a genetic test, right? Because it would look like Macrocystis.
It is pyrifera.
It is, yeah.
I'd love to answer your question, do a little research and answer it better. I'm sorry.
No, that's okay. I know this was a California Seaweeds talk, but I was just curious because I wonder if there's still like gene flow between the populations, or if they might diverge as.
I think the gene flow is pretty much stopped.
Yeah, okay.
Now. Except that, people move things around, so things can get blurry. I did some work in the Antarctic too. That's why, but I've never been to South Georgia.
Oh, cool. Oh, that's amazing. There was a leopard seal swimming in the kelp forest, and it was like the most amazing moment. Yeah.
Yeah.
Well, maybe I'll send you a follow-up email if that's okay. Thank you so much.
My email is cathyanmiller@berkeley.edu. If you send me a message, I'll get back to you on Macrocystis South.
Cool. Thank you so much.
You're welcome.
Yeah, that's great. If people have questions, go ahead and just ask to be unmuted or unmute yourself if you have a question for Cathy Ann right now.
Hi, Sarah. I have a question. This is Jim Rolfe. I am wondering, I tried to post it, and I obviously don't know how to post yet on Zoom, but
I don't either.
Wondering, I'm wondering about the introduction of non-native species into our local Mendocino coast environment, whether intentional by experimentation as our oceans warm, or by accident from coastal drift and the rest of the ocean's actions that are going to happen over the years. Can our guest comment on that, or what her feeling is? Are we
I don't think there have been many intentional introductions. I know in Europe, people moved kelps to grow them as mariculture, and they've spread the kelps into non-native habitats. But we haven't done that much, except through the oyster industry. The oyster industry brought shells over that had lots of spores in them, and many things have been introduced that way. So it's not intentional, it's unintentional introductions, but human mediated introductions. It seems to me in California, well, no, this is, whenever I try to make a generalization, I think of a difference. There seem to be more introductions in southern regions than in northern regions, but we just found a new introduced species in Oregon that we also have found in California. So I guess cooler waters welcome introduced species too. Am I getting at what your question is, Jim?
Yeah, my thought was, what we've done on the land in our history as humans, introducing new species that
Made it our own.
winds up,
Yes.
winds up biting us in the rear.
Yep.
I'm wondering, that concept applied to the ocean. I mean, the ocean is just, I'm only getting into what the ocean is. So as an
The
ecosystem,
ocean's magnificent. And
it is.
we have learned we can trash it, right? The
Yeah.
plastic story.
We are.
Yeah.
Yeah.
There's a lot of studies about the impact of non-native seaweeds and non-native invertebrates on native communities. And very few studies have given evidence that the non-natives are going to wipe out the natives. Usually they compete and join the community in some way. The one exception is Sargassum horneri. I showed you the meadow with the diver over it. That has spread like wildfire, and it's pissing people off because it winds up in their anchors and gets into their life too much. Okay, humans, boomers, settle down. The real problem is it actually taking space from the natives? And in some places in Baja California, Mexico, they're reporting that it is taking over what used to be kelp beds. So there is a fear that these introduced species can successfully compete, but it's not a given that they will be harmful. Most of them can't get big enough populations. Most of them are fairly small bodies. But when you get something big that's 6 or 7 feet tall, then you can start to really compete. So each introduction has its own story. We had all the tsunami debris from Japan some years ago, and there haven't been many seaweeds introduced to our knowledge from that. But we feel that there's probably been tsunami debris forever. And historically, Japanese wooden ships have washed ashore in California. So there's been introductions through time, human time and pre-human time. And it's the roll of the dice with every introduction. I think a lot of things are introduced and never make it, but they're introduced again and again and again, and something will be right, and they'll be able to hang on in a new environment. That's my take on it.
Yeah,
Right.
a number of things that you've spoken to with your knowledge and 40 years of being in this ecosystem give me hope, which feels good. I'm being
Glad.
eyes wide open right now, and documenting what we're seeing is inspirational and important. So
I think monitoring is one of the most important thing we can do. And I'm very happy that there are beach walkers along the coast. There is the intertidal monitoring system. Many universities are part of that, doing every year or every other year, counting intertidal species. There's some subtidal monitoring. I think we have less of a grip on the subtidal than we do on the intertidal because it's more accessible. But if we don't watch, we will not know what might be occurring in our own front yard. So
Exactly.
monitoring is extremely important.
And let's see, Larry, you have your hand up. Go ahead.
Yeah, I have a question, Cathy Ann. It's great to meet you. I'm actually a seaweed harvester up here on the Mendocino coast. And my question is, I've heard that Postelsia and Nereocystis are closely genetically related. And can you confirm, deny, give me feedback on how they're similar or what they share, anything like that?
They are closely related. The kelps used to be classified by their shape and by their growing zones, if they were split or not split. But now that doesn't work as well. And it's just on the basis of DNA sequence.
Mm.
So you do notice, however, that they both have hollow stipes. That
Mhm.
holds them together. Not all seaweeds have hollow stipes. They both form those blades from a little stipe-like base of the blade,
Mhm.
and that splits and forms more blades. So there are some morphological or form similarities, but they weren't originally thought to be related to each other. This is new evidence from the DNA.
Mm. Mhm.
So I have to tell you that I can't even remember the kelp families anymore because they've changed so much. And I've
Uh-huh.
lived quite a bit on the species level myself. That's just where I grub around. But the family, the constellation of the families has changed a lot because of our DNA evidence.
Mhm.
Am I getting to where you want to?
Yeah, I think so. Like I've been curious, for instance, do they share like settlement
Life
characteristics,
history?
or do they, like, for instance, when a spore travels out of a Postelsia and drops and attaches to the rock, is there some similarity in how Nereocystis
Yeah.
disperses spores or
Kelps
attaches or
are fairly similar, in fact. The kelps as a group, the order Laminariales, the order that contains all the families, have a very similar life history. So Postelsia forms spores on those blades that drip down the grooves on the blades and then hit the substrate, the mussels and rocks below.
Sure.
And then the alternate stage grows there, mates, and forms a new Postelsia. Similarly, Nereocystis forms spores on the blades. In the case of Nereocystis, it doesn't have those grooves that channel the spores like Postelsia, but the whole, I'm sure you've seen this.
Oh, sure.
Whole
The
piece
flora.
falls down to the bottom,
Yeah.
releases spores on the bottom where it's going to grow, and the alternate stage thrives, mates, and produces more kelps. So both of them deliver their spores to where they're going to grow their alternate stage, which is a hard bottom. They just have very different hard bottoms, don't they? One's intertidal, one's subtidal. Both are annuals. So that draws them together even more.
And how similar would, say, Laminaria seychellii be in that process?
Very similar, without a specific mechanism like Nereocystis abscising the tissue and dropping it to the bottom. Instead, Laminaria seychellii, I'm sure you've seen the dark patches of spores on the blade. They just swim away because it's an low intertidal kelp, and those spores have a nose for nitrogen, and they tend to be negatively phototactic. They go down to the dark. And so they dive down and sniff out nitrogen, which is mostly produced on the bottom by animals peeing and composting seaweeds, releasing nitrogen.
That's very cool. I didn't know that that
food.
nitrogen part of it is very cool. I didn't know that. I like
And they
that a
also
lot.
have a little eye spot, those spores, so that they can initially know what up and down is, light and dark, and then they dive down to the nitrogen gradient. So
Wow.
they're not
They're-
only wily, they're clever. They're
Wow.
brilliant.
And they're like motile and they're
They're
cruising
like
around.
motile, yes.
Yeah.
Well,
Very
have
cool.
you gotten the feeling that seaweeds can move? I mean, they're
Yeah. Oh,
not,
yeah.
they're not just like, they're not rocks. They're living, moving, breathing, photosynthesizing creatures that are ancient, have survived so long through the changing oceans, and will survive. I'd like, Sarah, another note of optimism. I believe that they will survive. It will be different.
Yeah.
That's
Well,
great.
thank you. That's great. And you know, actually, I just have to say something that's part of my enthusiasm for seaweeds, which is one of the things that I really love is in these high energy intertidal zones, how it allows so many species to persist because of all the movement and all the available photosynthesis and all the available nutrients with the water movement. And it's really something that's just so amazing when there's
But
so
another
much.
thing is the disturbance. Because
Exactly.
all that energy clears areas for new inhabitants too.
Well,
So
and I also think about how that movement creates a shift in available light. So it's not a
Yes.
static thing like a tree that's really creating a uniform understory light situation. It's
Oh,
actually
yeah.
the light is shifting. And so it's making it more available for understory species.
Absolutely.
Yeah. So anyway, thank you very much.
We'll take Mary Ellen real quick and then move on. Go ahead. You gotta unmute yourself.
I have one kind of little technical question. I would be interested in finding seaweed and pressing it. And I was wondering with your press, what happens to all the water?
The water gets soaked up by blotters. So you saw my press was different layers. There's cardboard to let air in through the corrugations. And then there's blotters. No one knows what blotters are anymore. I mean, that's before my time, like the stuff you put on your desk for your ink. But anyway, blotters, and then a piece of cloth to protect the seaweed and another blotter on top. And then you press them and change the blotters. That's the key. You have to take the blotters out, put fresh blotters, and press again. And this can sometimes take a week for thicker things, or for smaller things, a few days, depending on the ambient temperature, or if you have a fan or a little bit of wind. This is like 18th century technology, but it still works, and it's the way we do it. But you can cut cardboard of any size. You can use watercolor paper, any cotton rag paper that won't dissolve in water. You can put a seaweed, you can use any fine cloth, like an old sheet is good, percale. And for blotters, you can use newspaper if you have newspapers, because they will soak up water too if you don't have actual blotters. And then you just put those sandwiches, cardboard, blotter, seaweed, blotter, cardboard, blotter, stack it up, put a weight on it. Don't collect Postelsia. It's illegal, even if it's in the drift. But all the rest, if you're not in a specially conserved area, you can pick up off the beach and press. Do it. It's fun.
Do you throw your blotters away?
No, I have blotters probably from Sechel's time.
You just let them dry?
But we keep getting new blotters too. You can order blotters from herbarium supply places.
Thank you, everyone, for being with us on this evening. What a lovely conversation. And thank you, Cathy Ann, for sharing your knowledge.
I've
And
enjoyed you all. What fun. Thanks for the opportunity. Good night, everyone. Thank you.
Thank you.
thank you.